video is Unit 7, Learning Target 10 on an Intro to Mendelian Genetics. Gregor Mendel is considered the father of modern genetics. He worked as a breeder of pea plants, and he also bred bees, but his work with pea plants has helped science understand inheritance, and that's why he's largely considered the father of modern genetics. Now, in his work with pea plants, he normally would allow these pea plants to self-fertilize. And what is self-fertilization? This is when one plant provides both gametes, both sperm and egg, and it self-fertilizes. This is not asexual reproduction. This is still sexual reproduction because you still have male-female gametes and they combine for fertilization, but this is when one plant provides both. And he would oftentimes use plants that are called true breeding plant, and a true breeding organism is an organism that always passes down the same trait, a white flower that can only ever pass down the white flower trait. It makes white flowers, that's all it makes. So in order to mix traits, what he would do is cross-pollinate the plants. He would take the pollen from one plant, the male reproductive cells, and he would combine them with the ovary, with the female reproductive cells of another plant, and he would allow them to cross-pollinate. And in doing so, he would create what's known as a hybrid. Now, this is a pretty ridiculous hybrid, and so we're not talking about mixing horses with cats or anything like that, but what we're doing is we're talking about like mixing a white flower with a purple flower, or a tall plant with a short plant, in order to create a hybrid, which is just a genetically dissimilar organisms, or a cross between two genetically dissimilar organisms. Now, what is a trait? A trait is a specific characteristic that varies from one individual to another. Traits are things like, well, in pea plants, it's seed shape and seed color, and there you see them. And in humans, it can be stuff like this, like, are you a tongue roller? Do you have attached earlobes? Do you have freckles, or Morton's toe, which is where your middle or second toe is longer than the big toe? Kind of strange. But anyway, these are different traits that humans have. A trait, as you can see, can have multiple expressions. The trait of eye color, for instance, has multiple expressions in humans: blue, brown, green, other. Right? Hair color, height, all the different kinds of traits that you can think of have multiple expressions. So when Mendel was doing his crosses, he would label the generations of the crosses that he was doing, and the first cross was always the pea generation, and this was always parents of a true breeding type. And so he would take true breeding purple flowers and true breeding white flowers, for example, and this would be the pea generation, the first generation. Well, the offspring of that parent generation were the F1 generation. Then he would take those F1s and allow them to self-fertilize, and then they would create their offspring, which are the F2 generation. Now, we normally just refer to this P, F1, F2 when we're talking about plants. We don't usually talk about humans this way. We use things like pedigrees or family trees in order to talk about human traits, but because humans aren't self-fertilizing organisms, and so the F1s can't self-fertilize to make the F2s, but you kind of get the idea. But this is terminology that Mendel would use and that we will be using in class. Now, what's important to understand about traits is traits are coded for on the DNA. DNA expresses itself by making proteins. So that means the proteins that your body makes will express themselves by giving you blue eyes or brown hair or height or whatever you have. And those genes have different versions. We each have one chromosome from mom, one from dad, and they both gave us a different version of that gene. Those gene versions are called alleles. And so here you have a homologous pair. Remember homologous pairs. This is an example of a homologous pair here. Well, in this case, the gene for flower color is located on this chromosome. One version of that gene is purple. The other version of that particular gene is white. And so which one is going to show itself? And that brings us to the next idea, which is Mendel's principle of dominance. Very important to understand in Mendelian genetics is the principle of dominance. And Mendel's principle of dominance was this, that some alleles are dominant, some alleles are recessive. The dominant alleles will always express themselves, and the dominant alleles will always mask the expression of the recessive alleles. And so if the dominant allele in flower color is purple and an individual has a purple allele, then you will always see purple. You won't see a mix between purple and white. You'll see purple because white cannot express itself when a dominant allele is there. The only way it can express itself is if there are no dominant alleles. So if there are two recessive alleles, for instance, we all have two copies of a gene, two alleles. Well, in this case, here, there are no dominants, and so the white can express itself. Well, there are three different terms we're going to use for this. This first one, where there are two dominant alleles, they are the same, so we're going to call this homo means same, zygous for zygote, homozygous dominant. They're the same, they're both dominant. Well, the next one then would be homozygous recessive. And so it's only when the genes are homozygous recessive that the recessive allele is expressed. So then what's the last one? It's got a white and a purple, and it shows purple. So we would say this is heterozygous. We don't say heterozygous dominant or heterozygous recessive because it's just heterozygous. Now, where we go from here? Well, Mendel, what he did is he took true breeding plants of a dominant, let's say green pea pods are dominant over yellow pea pods. Well, he took true breeding green pea pods and he bred them with true breeding yellow pea pods. True breeding green pods will only make green. True breeding yellow, because yellow is recessive, will only make yellow. So here you have, if you want to do with Y's, big Y, big Y, and over here you have little y, and he crossed them together. Well, what happened? Where'd the yellow go? Remember, it is masked by the dominant allele. And so then what he did is he allowed those F1 generations to self-fertilize. And what happened? Well, the yellow came back. Why did the yellow come back? Well, that's Mendel's second principle, which is the principle of segregation. And Mendel's principle of segregation says this, that the alleles of a gene separate during gamete formation. During what is gamete formation? Meiosis. So during meiosis, the alleles of a gene will separate. Or another way to say that, the different versions of a gene have an equal chance of being inherited. And so if you have both the dominant and the recessive allele that you're able to pass on, you have an equal chance of passing one or the other. And so in this case, we end up with a yellow pea pod a quarter of the time. Here's another example of this. True breeding tall plants with true breeding short plants, homozygous dominant tall, homozygous recessive short, mix them together. Of course, you get 100% tall. However, each one of those tall plants has the recessive allele. And so when you mix them together, what happens? Part of the time, those two recessive alleles are going to combine. And this is the principle of segregation. Let's look at this way, not that way, this way. So this is your genes for this particular trait, whatever this trait might be. And you're able to give the little a as equal as you're able to give the big A. Another way of thinking about this, let's go back to our meiosis. remember from meiosis, we would draw these little chromosomes, and that's a homologous pair, right? Well, on this particular homologous pair, let's say that the gene for pea plant height is located on this homologous pair, and that gene is located right here. And this particular individual has one dominant allele and one recessive allele. Well, what's going to happen when interphase occurs? Well, the DNA is going to replicate, right? And so we're going to say this individual is going to have two big T's now because the red one's going to replicate, and two little T's because the blue one's going to replicate. Well, what's going to happen next? Meiosis 1. During anaphase, the homologous pairs separate, and so what goes over to the left? The two big T's. Because red separates from blue, not red from red. And the blue goes over here. And then what's next? Well, we have an equal chance of getting big T as we do little t, or 50-50. Well, what if both organisms, so what's the chance that the little t's are going to get together with two organisms that look like this? Well, there's a 50% chance of it happening here, right? What if we did this again? There's another 50% chance. And so what's the chance they're going to have an organism that's two little t's? A quarter of the time. Didn't we see that? We did see that. 1 out of 4. Well, what's a better way to show that in a very visual way that is easier than doing multiplying fractions, which I think multiplying fractions is particularly easy, but if you're not good at that, we can use a diagram called a Punnett square. And in this Punnett square, we can put our big T, little t on the side, we can put our big T, little t on the top, and we can show that 1 out of 4 times the little t's will combine. And we'll talk more about this in the next lesson.